All right, hope everybody can see that. Can you confirm that you can see the presentation?
Yes.
Great, all right. So, as you just introduced, I'm from Nkt Photonics, and I'll be talking a bit about this type of light source called a supercontinuum laser, and what it's used for, and how you could potentially use it in your work. So I'll go over the following topics today. I'll first introduce to who we are and what we do at Nkt Photonics. Then I'll narrow it down and talk about the white light lasers or supercontinuum lasers, and what they are used for. And finally, how a bit about how the technology behind these lasers work. So if we look at, for a start, at what our lasers are used for, it's a wide variety of things. We have several lines or product lines of lasers, and they're used in everything from medical early disease detection. I'll actually talk a little bit about that today. They're used in characterization of components that go into consumer electronics, such as smartphones. We have a lot of activities that is aimed at the field of quantum computing and AI, as well as cancer and stem cell research. I'll also talk a little bit about that today. In renewable energy, our lasers are used typically for windmills to improve efficiency of windmills. That is actually something we do with laser. And space exploration, so we have lasers in space conducting scientific experiments as we speak. Our goal is to provide solutions for innovators. We are ourselves innovators. We have to be for the type of lasers that we produce. I'm sure that will be very clear at the end of this talk. But we see ourselves mainly as enablers that enable you guys to be successful in your work by using our technology. All right. So if we become a little bit more specific about what it is that we do in these areas of research. In cancer research, we have for many years been the default light source in the fluorescence lifetime and STED type microscopes, providing a very exotic light source that enables these type of microscopes to function. I've written here cystic fibrosis, which is a lung disease that greatly reduces the average lifespan of a person. And we are helping researchers with our technology, creating endoscopes that can monitor and diagnose these diseases to improve the life of people with these diseases, but not just in lung diseases and this type of endoscopy, but in many types of endoscopy. And chances are that if you had eye surgery with lasers, there's a pretty good chance that, in fact, our laser was the ones that was used in there. So as you can see, quite a variety in the applications just within the medical solution space. Within industry, as I mentioned, our lasers are used for renewable energy. This is not the topic of today. What I'll be talking about today is neither the how the lasers play a role in renewable energy or how we enable safe tunnels and metros around the world, but I will talk about how lasers can be used to quality control products such as food, pharmaceuticals, or plastics. And finally, we have aerospace and defense applications. I won't be talking over these. These are mainly related to high power laser systems that are used to protect airports and critical infrastructure from drone attacks. So if we have a great overview of exactly what kind of lasers enables this, it's all at the core of it based on our fiber technology, which is these photonic crystal fibers. I will get back to what a photonic crystal fiber is and why it is so important to master this technology to be able to enable these laser technologies. And this is both regular fibers that guide light, but it's also fibers that amplifies light to very high powers with pristine beam qualities. And from these fibers, we can make various types of lasers: supercontinuum lasers, ultra-fast lasers, and single frequency lasers. What I'll be focusing on today is the supercontinuum lasers. If you need information about any of the other things, feel free to take contact to our representatives in Japan, Sevensix. Finally, we have a line of sensing systems, which is the ones that are used to monitor critical infrastructure. We'll be talking about that today. So a great overview of our history. In Nkt Photonics, we are in fact a quite old company back from 1891, where our founder electrified most of Scandinavia with power cables. Up until today, so starting with power cables up until today, now we are the foremost technology in the company now is these fiber technologies, different type of fiber. We are located a bit around the world, centered around Europe, as you can see, with our headquarters in Denmark, where I go to work, but also with manufacturing sites in the UK, in Switzerland, and in Germany, and most recently with a production site in the United States. As I said, we are innovators ourselves, evident by our large patent portfolio that protects and ensures our technology, which I'll be talking about today. All right, so what kind of problem are we solving here? I think this illustration shows it quite well. If you are on the lookout for several laser lines and you want to use them simultaneously or in sequence, but along the same beam path, you typically would have to combine several types of lasers. A typical laser only has a single wavelength. So if you want multiple colors, wavelengths out of a laser, you would have to combine them somehow, which can be a tedious, laborious process and introduces a lot of complexity in assembly. And what a supercontinuum laser does is it takes the functionality of an infinite amount of lasers in the visible and near IR and combines it into a single, very easy to use output fiber laser. So I've said supercontinuum a couple of times. So what exactly is a supercontinuum? A supercontinuum means a spectrum that covers a very wide range of wavelengths. As you can see here, going down from 400 nanometers, which is the beginning of the visible spectrum, through the entire visible spectrum, which ends up around here, around 750, and all the way up to 2 micron. All of this out of a laser light source. So this is typically what you would expect of maybe a lamp. But in many applications, a lamp is not really feasible, and that's what I'll be talking a little bit about today. What this graph also shows is a bit of the differences between systems. So as you can probably see, there seems to be a trade-off between the wavelength, the shape of the spectrum and the intensity of the spectrum, and its starting wavelengths. This is the main difference between the spectrum. We define down here the 400, how ultraviolet does the laser need to go. If it's to be as ultraviolet as possible, it can't at the same time have the highest possible spectral density. All right, so if we could boil all this down into what are the 5 main features of a supercontinuum laser? It is broadband, as I just showed you. It is bright. What I mean by bright is it has, just like a laser, you can focus it down to a very intense spot with a lot of power in it. It is single mode, which is what allows you to focus it down to a single spot. Unlike a light bulb, which cannot be focused down to a single spot, it can simply be projected onto it, but you're not going to burn a hole through anything, even with the best magnification of a light bulb. It is tunable. I'll get back to that. That's not immediately clear how that works. And one thing I haven't mentioned is this is in fact also a very fast pulse laser, which opens up to a whole area of applications that you would not be able to do with a lamp or an LED. And I'll get back to that in a second. So you can, in many ways, see this as a Swiss Army knife of light sources. It combines a lot of the features of lasers that makes lasers attractive in applications, and combines it with the versatility of LEDs and light bulbs, which of course means that the area of applications that these are used in is vast, a huge number of applications. And I've just here written some of the techniques that you would typically find a supercontinuum laser in. So if your technique is somewhere on this list, chances are you should be considering a supercontinuum in your setup. So regardless of what kind of characterization or data collection you're doing, you're probably looking at some sort of specimen. This could be a biological sample as shown here, but it could also be a food sample. It could be a plastic. It could be anything really. And when you do optical characterization of something, you shine a light on it from a light source, and looking at what comes through the other end, you can say something about what light disappeared in the process or was generated in the process. Or you can look at what is scattered out of a diffuse sample, which gives you another set of information. I'll talk about optical coherence tomography as one example of this. And you can look at what is emitted as light from different wavelengths than the ones you expose the sample to. This is the world of fluorescence. And what a supercontinuum laser allows you to do is not only do this at all wavelengths, but also do this in a much more sophisticated manner, using pulses to estimate, for instance, how long does the fluorescence last in such a sample. And just because it's a laser and I say you can focus it down to a small spot does not mean that you can only use it on small samples. In fact, you will find these lasers used at almost any length scale, with it's used as a solar simulator, illustrated on the far right here, all the way through optical component characterization, fiber characterization, fiber characterization down to metamaterials, quantum dots, and so on, all using one of three or several of these traits of a supercontinuum laser. So one example of this, actually I'm going to go through a handful of examples now, just to show you how it is sometimes used. One example of this is in the world of perovskites, where maybe saying that perovskites here is a material that is a contender for solar cell materials. So if you are used to silicon solar cells, they have an approximate best case efficiency of around 27%. It's not theoretically possible to harvest more light from the sun with a silicon solar cells than thereabout. What perovskites do is allowing solar cells to be manufactured with much higher efficiencies at very low costs. So a lot of research is going into perovskites. But it's a material system that allows for a lot of customization, which means that, and it is a system that has wavelengths of interest across the entire visible and near infrared region, which is where you want to harvest the solar spectrum. So depending on how these perovskite materials are designed, they will absorb light at different wavelengths, and the electrons that is what you want to harvest in these materials have different lifetimes, which is something you're very interested in researching, as you want the electrons to be living long enough as an excited electron in order to harvest it and get any use of it. If the electron disappears the moment it is excited in the material, it is of no use. So not only do you need to do spectroscopic analysis of these specimens, you also need to understand the dynamics, the time dynamics of these materials. And this is where supercontinuum comes very much in handy, because you cannot take a light bulb and pulse it on a picosecond scale. And you cannot take a typical laser and tune it across all these wavelengths. A supercontinuum laser is the only light source that allows you to do both of these things that are so important in solar cell research. So what you actually see over here is on the top a range of quantum dots prepared as perovskite quantum dots prepared to absorb light and emit light at different wavelengths. And what you see below is the difference in decay lifetime of these quantum dots. So exciting the quantum dot with light and seeing how fast does it take for light to return. So this technique of creating fluorescence and looking for the fluorescence lifetime is something that we will also see when we move into the world of bioimaging. So this is all in order to find the candidates with the best lifetime and the best light absorption. With a supercontinuum, you can make fully automated setups. You don't have to change your light source every time you change either the wavelength or your pulse your pulse repetition rate. This can all be done with a supercontinuum list. This is just an example of a setup that has been created with a supercontinuum that allows for full characterization by the click of a button, and out comes characterization of the material in question. Okay, so as promised, this fluorescence lifetime is also relevant in the world of bioimaging. So quickly on what fluorescence is, what I have here a picture of is actually phosphorescence, but for all intents and purposes, this is just a slower version of fluorescence, which is why you can take a light bulb, excite phosphorescence, turn the light bulb off, and see the phosphorescence decay on the second to minute scale. With fluorescence, it's the same concept. You excite a specimen to an excited state, and through a decay, you see emission that is specific for that specimen. It could be a molecule, or it could be a perovskite crystal, as we saw before. But this decay time is on a nanosecond scale, just as we saw before, which means that you need something that can turn the light on and off, essentially pulse the light on the time scale that is shorter than this. And a supercontinuum laser typically has a pulse duration in the picosecond time scale, which means that you can excite and look for the emission get an idea of what the lifetime is. And why is this so interesting? Well, it turns out if you take a single fluorescent molecule and you put it in different environments, it will affect how long it stays in its excited state, which allows you to, with a single fluorophore at a single wavelength, to create contrast that is distinguished between whether this molecule is sitting, say, in a cell membrane, or it's sitting inside a living cell, or it's sitting outside the living cell, just by looking at how fast light is emitted after it's been exposed to a pulse. So you could now imagine having several of these molecules that allows you to use fluorescence. And for each of these molecules, you can look at the lifetime and get different colors based on your lifetime. And this is what Leica does with our supercontinuum laser, where the supercontinuum laser is the driving light source in it. So what you see here on the right is an example of what value it gives you to have information about lifetime and different fluorophores. A gray picture does not really tell you about what is the different tissue, and are there areas of interest within that tissue other than structural information. But what you get with lifetime and what's called fluorescence emission excitation spectroscopy is a whole lot more information, as you can see here. So what the colors here mean, they represent both different fluorophores. Fluorophores are the molecules that emit light, but also represent different lifetimes of that fluorophore, greatly increasing the amount of information that you get about whatever it is you're looking at. And it doesn't have to be restricted to solar cell materials or biological molecules. It could be anything that emits light in the visible or near infrared. Another example of fluorescence, how fluorescence is used in microscopy, is in light sheet microscopy. A light sheet microscopy is a microscope that uses a sheet of light. The light sheet is generated by focusing light through optics and generating a very, very thin micrometer thick sheet of light at a wavelength that will allow excitation of one of these fluorophores. Then you take the specimen, which is typically small, say rat brains, or anything like that, has been stained with these fluorophores, and you pass it through the sheet as you monitor the fluorescence. And what that allows you to do is create these amazing three-dimensional images with different tissue stained. This is an example of a mouse embryo with two colors of fluorescence, yellow and green, showing different where different tissue is in a completely 3D. This is not actually a 2D image. This is a 3D image. We of course only seeing the 2D representation here. Another example could be a zebrafish here with a count 1, 2, 3 different fluorophores used. If you were to use any other light source for this, either you would use a laser, which would only allow you to use one fluorophore, that be one of these colors, or if you were to use a broadband incoherent light source, you would get a much worse resolution. So by combining both wavelength tunability and a light source that can be focused down to a small spot, you get the best of both worlds. All right, so moving on, we're still staying within bioimaging. Optical coherence tomography is another technique that greatly benefits from a light source like a supercontinuum laser. It is much like ultrasound, but a higher resolution and shorter range. So ultrasound typically is used in a range of about 10 centimeters with millimeter resolution, where optical coherence tomography is used on a millimeter scale, but with micrometer resolution, which is why it lends itself very nicely to study tissue or thin films if used in an industrial context, where you need to look at something with micrometer resolution through typically dispersive medium. What you see on the right here is an example used to diagnose melanoma, that is skin cancer, by looking at the at moles on the skin. These are actually examples I'll get back to from our partner at Damay Medical in France, who has devised a setup that allows them to identify skin cancer cells just by looking through the skin from the outside. Finally, within optical coherence tomography, supercontinuum also allows the user to introduce what we call multimodality. And I'll give you some examples in a moment what multimodality means. So if we look at what an OCT system does, it's as you can see, quite a simple interferometer. That is, light is entering on the left and is split in two. One half of the light is sent to a reference. The other half is sent to the specimen, in this case, skin. And we'll see what it then does. So I hope you can see the little animation here. Either the reference arm or the distance to the specimen is changed, which allows you to create this a three-dimensional scan, of whatever it is you're looking at. So, as mentioned, our partners in France, Damay, has devised this handheld device that allows you to very easily point and shoot at a site on the skin and test for melanoma in just a few seconds. So what really makes supercontinuum lasers unique for this type of application is evident by this graph here. What you have on your y-axis here is the resolution as a function of the bandwidth on the bottom here. And each of these lines represent a wavelength region. So at the bottom here, you have the wavelength region around 600 nanometers. 600 nanometers, for those who are not used to thinking in terms of wavelength, is roughly orange light. 800 nanometers is slightly infrared, and 1000 nanometers and 1300 meters are increasingly infrared, so longer wavelengths. As you see, the shorter the wavelengths, the higher the resolution, but also the wider the bandwidth, and that is how incoherent the light is, the higher the resolution. So for the highest resolution, you need the highest bandwidth and the shortest bandwidth. This is only really possible with a supercontinuum laser. Sometimes, though, you do prefer to use longer wavelengths, and that is for reasons of depth of penetration, so how deep into a given tissue or sample that you go depends on the wavelength in an inverse manner. So if you want the highest resolution possible, you can get that at the 600 nanometer mark, or if you want the deepest penetration into the sample, you go for a longer wavelength. With the supercontinuum laser, you don't have to choose. All of these are available at the same time. But there are plenty of ways to make optical coherence tomography work. One thing that also sets the supercontinuum laser aside from any other type of light source is the multimodality. And what I mean by multimodality, it allows you to do additional characterization in addition to the tomography. So while you're collecting a 3D structural image with OCT, you can at the same time introduce other wavelengths and conduct spectroscopy or fluorescence lifetime, as we just saw a few moments ago, or any other thing you might want to do at the specimen with the light source. You can do any type of spectroscopy on top of doing optical coherence tomography. An example of this is Optician, a company in Chicago who looks at how oxygenated blood is inside blood vessels. So not only do they look at the structure of the blood vessels, they also look at how oxygenated, how much oxygen is in the blood inside the vessels. This is called angiography. And what they do is they look at essentially this. This is the different. This graph shows the difference between oxygenated blood, HbO2, and deoxygenated blood, Hb. You can see there's a couple of places here around 500 nanometers, and again around 600-700 nanometers, where there are spectral fingerprints that allows you to distinguish between whether the blood is oxygenated or not. So this is what they do. They take a fundus image. This is an example of an OCT image through the eye, and where you can see the blood vessels flowing in and out of the eye. It's not immediately clear from this image to the far left what is going in and what is going out of the eye. But on the next picture, they've taken this additional wavelength at 500 nanometers and looked at what is the oxygen content of the blood in each of these. And from this, you can see that the blood is flowing in through the red and out through the blue, deoxygenated. So I'm just going to show you a little picture here of this. There you can see the image being collected. This is the OCT retinal imaging in the visible, completely 3D mapping the eyes. And this is important because all of this tells you information about the state of the retina. And what you may not think about daily is that the neurons that are present in your eyes are actually an outgrowth of your brain. So not only does it tell you about how well oxygenated your retina is, which in fact can tell you something about diseases such as diabetes, but you can also get information about what is the state of your brain, because if something is going on in your brain, chances are you can see it, or there will be evidence of it in the back of your eye. So eyes are a very interesting place to look for precursors for diseases, that is early disease detection, which is what another company called Optina is doing. So what they're creating is this hyperspectral camera that uses the spectrum of the supercontinuum to look for precursors for Alzheimer's disease in the retina of the brain. So they're collecting images at all these different wavelengths and using advanced algorithms to identify starting symptoms of Alzheimer's disease. There you can see collected images at different wavelengths. And again, looking at the state of the vessels inside the eye. So that's just some examples of what you can do with an OCT. But what you could also imagine looking at is you could look at the thickness of films in a production. We have customers who are looking at that. Or any place where you want micrometer resolution of maybe a surface that you need to do quality control of. So this is where I believe this is what you guys are good at. We're just here to provide you with the tools to do it. Okay, so now mentioning hyperspectral imaging, maybe a quick note on what that is and where a supercontinuum laser is particularly useful. So what you see here are 4 different ways of doing hyperspectral imaging. On the far left, we have point scanning. So that's a simple spectrometer, and then you scan the laser line over the sample. This is a cheap and simple way of doing very fast hyperspectral imaging, and is what some of our food sorting customers are doing in what's called I'll get back to that in a second. Next up is line scan. So here, instead of having a simple one-dimensional spectrometer, you have a 2D array where you project your spectrum onto. So just as before, each line here represents a spectrum, and each column represents a point on this line. So this is what you would typically call a tube-fed sorting, where you move a conveyor belt through a line and collect a spectrum for each point along that line on the conveyor belt to create a full 2D image of everything that passes through your conveyor belt. This is a typical laser collection method used in sorting of foods. And next up, there is a few other types of hyperspectral imaging that are more or less useful. I'll be focusing on the first 2 here. The first one is point scanning. So point scanning, examples of this is flow cytometry, where you are sorting cells. So of course, the beam characteristics are important. You need the beam to be small enough that you are only illuminating one cell at a time. And at the same time, if you these sorters typically work with several wavelengths, so you need something that is a laser that can handle several wavelengths, which is why supercontinuum laser lends itself so nicely to this. But it could also be something completely different, which is channel-fed optical sorting, which is what you will typically find in knot sorting or pharmaceutical solid formula sorting, and anything to that kind, where you have your samples passing through in a line. One example of this, I can't reveal the name of the customer, use such a channel-fed sorting method to greatly improve the throughput of their sorting facility, sorting their food specimens into different grades to be used for different types of products, which is something you can do spectroscopically. There are fingerprints, typically fingerprints both in the visible that tells you something about the color quality of your food, but in the near infrared, you can at the same time look at stuff like sugar content by looking at the spectral fingerprints of the samples. Another area where this is relevant is, of course, in plastic sorting, where the color typically reveals nothing about the plastic itself. You can have all sorts of plastics, all colored red. And how do you actually sort these and distinguish these from each other? And at the same time, if you have one type of plastic, it could be colored in all sorts of colors. So how do you distinguish those from each other? Which is where a laser like supercontinuum comes into play, excuse me, which allows characterization not only of the color, but also the content of the plastic. So very much like the food sorting, but completely different. So in conclusion, I just want to say, almost regardless of what it is that needs sorting, as long as it has some sort of spectral information, you can sort it using a supercontinuum laser. Okay, so that's the application examples I prepared for you. If you have any questions for those, I mean, you're all free to interrupt and use the or use the chat function, or otherwise, I think we'll have some questions at the very end. Next up, I just want to say a few words about what exactly goes on inside one of these supercontinuum lasers. At its very essence, it's very simply made by taking a pulsed laser and shining it through a nonlinear material, and out comes this pulsed supercontinuum spectrum. So a pulse laser through a non-linear material, and out comes a supercontinuum spectrum. This is very simplified. There's more electronics controlling this process than there is optics inside it. So if we then have a look at what happens to the spectrum after it's generated, I've been saying this is a broadband spectrum, but most of the time you need maybe one or a few wavelengths at a time, which is where our line of accessories and filters come in, which takes the spectrum and sorts it in an automated fashion. So turning a broadband laser into a tunable laser or into a multi-line laser is something we can do as well. So in the very beginning, I mentioned that this is based on photonic crystal fibers. So maybe a few words about what a photonic crystal is. A photonic crystal can be either 1D, 2D, or 3D. In nature, we actually find these photonic crystals, which is materials that get their optical properties from their structure, more so than from the material itself. So example of this is if you look at butterfly wings, when you get these very luminescent colors, the colors are all generated or all reflected from the same protein that makes up the rest of the wing. But by changing the microscopic structure of the protein in the wings, the butterflies and other animals can produce different colors. So this is what you see on the left, an example of a 2D photonic crystal, which is a crystal that is periodic in 2 dimensions. Could be this the colors in the wings of what's called a peacock, which is an example, as you can see down here, of a 2D arrangement of the proteins in the wing. And finally, there's also in nature examples of 3D photonic crystals. An example of this is this gemstone here. It's an opal that's typically found in Australia, which consists of microscopic silica spheres. And depending on the size and the distance of these silica spheres, it generates different colors in reflection. Our photonic crystal fibers are an example of an artificial 2D photonic crystal. It looks like this. So as you can see, it is a periodic structure in a hexagonal pattern created by introducing holes into a glass structure. So this is glass with air holes in it. And the center, the guiding core of these fibers is created by introducing a hole, or removing a hole where there otherwise would be one. So light is guided in the core here. And the whole fiber is defined simply by changing the pitch, that is the distance between the holes, and the diameter of these holes. By changing these two parameters, we can create all these different spectra that you saw in the beginning. And it allows us, first and foremost, to make these supercontinuum lasers that would be impossible to make if we did not have this technology developed. So this is the core of our technology, and what does that produce? So I've here compared a supercontinuum laser, a Super K, with a regular lamp as an SLED. And what you see on the bottom here is the wavelength coverage that we have from 400 nanometers at the UV up to 2,400 nanometers well into the infrared. Notice this is a logarithmic scale. So each of these ticks represent a factor of 10. So if you look at how much power you can actually focus down to a spot or a line or anywhere where you want to control the light and not just shine it everywhere, you get several orders of magnitude more intensity out of a supercontinuum laser than you do with any of these other coherent light sources. And this is a very key point. You get a lot more signal, which for many means a lot higher throughput with a supercontinuum laser than with any other incoherent light source. A few words about the filters I mentioned a little while ago. So if the entire spectrum is not to be used all the time, but some of the spectrum, some of the time, we have this wide range of tunable filter systems. That is filters where both the bandwidth or the wavelength can be changed in an automated and sometimes very, very fast fashion. Yeah, I'm not going to go into details about what each of these filters do. They are different in the way they control the light. You have a filter on the very left that allows a single line to be tuned both in its bandwidth as well as its absolute wavelength. The second filter allows you to pick any 8 wavelengths out of the spectrum and move those around completely automated. And the next one is a very pristine, very narrow line laser filter that allows tunability across the entire range of the supercontinuum laser, and so on and so forth. One thing to mention also is all of this is delivered as a simple fiber out of the box. It takes less than a minute to get this system up and running, and is very stable and runs for thousands of hours, as I'll show you in a few seconds. So in conclusion, I hope it is now clear that we're dealing here with a light source that is not only allows for pulsed operation for some of the applications we were talking about. It's first and foremost a broadband light source with filters allows you to turn it into a tunable single line laser or a triggered laser or even a multiple line laser with the right accessory. These all lend themselves to different applications. So one or more of these aspects of the laser will be relevant to you depending on what type of applications you're looking at. And they come in all sizes and shapes depending on where they are to fit in. We have a line that is focused on industrial applications with up to 8 watts of focused power. That is typically more than enough to burn holes and things. So don't compare them to an 8-volt light bulb. This is a lot more powerful than even 1,000-watt light bulb. On the scientific side, we have very versatile platforms that can be used for many different experiments. We have not just discovered this. We've been doing these types of lasers for almost 20 years now. We constantly have 60 lasers just running lifetime tests to ensure that these lasers are robust and will survive for many years in their hands, even in the hands of 24/7 operations. And I think that's it. And I'll hand the word back to the organizer. Thank you very much.
東さん、どうもありがとうございます。